Today we're going to be talking about ultraviolet light signaling in animals.
So what is UV light? Ultraviolet light is a type of electromagnetic radiation with a wavelength ranging from 10 to 400 nanometers. This is on the lower end of the visible spectrum. We're familiar with UV radiation in the form of sunlight causing sunburns, or in black lights, or even in tanning beds. However, we cannot see it because we have trichromatic vision with only 3 cones for color, these being our S cone, our M cone, and our L cone. Some animals can see UV light, however, meaning that they have some sort of specialized structure in their eye that allows them to do so. This would be the extra UV cone in their retina.
There are many animals out there that have the ability to see UV light. For example, reindeer rely on UV light to spot lichens, a type of plant they eat. The sockeye salmon also uses UV light to spot their food on the ocean floor. In addition, many insects can see UV light, such as the bumblebee that can see the black-eyed Susan flower by its unique bullseye pattern that was only visible by UV light. So these animals just discussed are able to see UV light, which helps them find their food in challenging and busy environments. However, other animals are able to signal and communicate to other animals that can also see UV light. These animals are often, but not always, will have UV reflectance directly on their own body. Some examples of animals that are actually able to communicate with UV light are the cabbage white butterflies, which are thought to have the widest visual range of any animal. These butterflies can use UV markings to pick out healthy mates. Other species of butterflies use UV patterns that make them appear similar to predators while differentiating themselves to potential mates. And then the jumping spider uses UV markings to excite the opposite sex's mating interest. And then finally, a very large variety of bird species have UV markings, and some use these for sexual signaling, and some have also been found to feed their young based on how much UV they reflect.
The first study we will be discussing looks at multimodal signaling and how UV reflectance predicts male breeding success better than pheromones in the butterfly Coleus erytheme. Before we move on, we should understand what the goals of the researchers were with this particular study. They set out to understand the relative importance of pheromones and UV signaling in courtship. They asked the questions, have multiple signals been developed to either 1. Give more information about the different traits that the signaler has, or 2. Work as backup signals for a single trait. And then they also asked, which predicts male mating success more accurately, pheromones or UV signaling? A little background information. Females of this butterfly species often observe one or more male secondary sexual traits when they're trying to find a mate. Earlier we discussed how this study is going to be looking at multimodal signaling, and all this means is that signals can either occur over a single or multiple modalities. Examples of this could be chirp rate and carrier frequency that would both be auditory, or four-leg tuft size and vibration patterns, which would be tactile and visual. And then this all gets brought back to, did multiple signals get adapted to either give more information about different traits or work as backup signals for a single trait? Continuing with the background information, Coleus erytheme send visual cues via their UV reflecting scales and olfactory cues via lipid-based cuticular hydrocarbons that are more commonly known as pheromones. And we know separately that these both encourage male mating success, but no studies have made an attempt to evaluate which signal is more associated with success in the field. Beginning with the methods, the researchers actually reared their own virgin Coleus erytheme females from eggs. They kept these eggs at 4 degrees Celsius for up to 5 days, and then marked on both of their ventral hind wings with red felt marker to help ID. And they only used the orange-yellow phenotype because the alba phenotype is, one, very hard to identify, and then also shown to have different preferences in males. For the mating trials, one singular female would be released into a field of male butterflies, and a mating attempt was recognized when a male was within 0.5 meters of that female. It ended when the pair was either mated or the male just left. The unsuccessful males were captured, and the successful mated males were separated before they could exchange seminal fluids. Then, the accepted and the rejected males were both freeze-killed at negative 10 degrees Celsius. From here, their UV reflectance, pheromones, body size, and age were all measured. On the right here, you can see how the researchers classified wing condition on a scale from 1 to 9, with 1 being no scale loss and 9 being few scales left with severe tattering. To accurately measure the pheromone levels on the wings, 3 out of 4 of them were taken and made into a solution that was put into a varying gas chromatograph mass spectrometer. Statistical analysis saw the use of t-tests, the AIC, walled tests, all being done under a 95% confidence interval. And the UV reflectance was measured using 3 different methods: reflectance spectrometry, directional reflectance spectrometry, and whole wing UV image capture. The researchers found 3 main results. 1. That females prefer to mate with younger males. 2. That the UV reflectance and pheromone traits covaried with age. And 3. That the brightness of the UV reflectance was the strongest predictor of male courtship success. Here we see the results for the wing length, the 3 UV tests, as well as the pheromone tests for the rejected and the accepted males. The accepted males tended to be younger than the rejected males, giving them higher scores on mostly everything except for the pheromone C27, which is not an honest indicator because it does not diminish with age. Here we see the summary of the results for the principal component analysis. It was conducted on all the raw data with population equaling 96. Loadings greater than 0.7 were highlighted for emphasis. Finally, this graph shows the mean reflectance spectra of accepted, represented by the solid line, versus rejected, represented by the dashed line males, measured using directional reflectance spectrometry. The researchers concluded that multiple signals do in fact act to increase signal clarity about the same trait, with UV reflectance appearing to be more important to female coleus erytheme when choosing a mate. This could be due to UV brightness being an indicator of male health. During the transfer of seminal fluids, the male transfers proteins to the female via the spermatophore. We know it is a costly process to produce these proteins, so this increases the female's fitness. This is also what's known as material benefit. We know other species, such as grosbeaks, rely on UV reflectance as an indicator of male health. Therefore, more research should be done on the extent UV reflectance relays phenotypic and genetic information about male coleus erytheme mates. Future research should also be conducted on whether pheromones are an honest indicator of age, which remained untested at the time of this article's publishing. They should also keep in mind that pheromone C27 is not an honest indicator of age.
The second study we will be discussing is about how ultraviolet signals in birds are special. The goal of this experiment is to examine if there is UV reflectance on different species of birds and if this reflectance is associated with sexual signaling techniques. The researchers asked the following questions. Is UV reflectance plumage more prominent in body areas used in courtship displays? And are areas of plumage that are used in courtship displays more colorful in general, meaning is there anything significant about the UV reflectance plumage, or do birds just like other birds that are just colorful, even if they're non-UV colors? First, we need to understand how birds can see UV light. Birds have tetrachromatic vision, meaning that they have 4 cones in their retina. These include some of the same cones as humans, so your short wavelength cones, your medium wavelength cones, and your long wavelength cones. But then they have one additional special UV sensitive cone. This is illustrated as a comparison with the human vision in the image to the right. So before we move on, we'll want to just define some things to make sure we're on the same page. So first, what is UV reflective plumage? It's exactly what it sounds like. It's the feathers on the bird that are able to reflect UV light. Therefore, other birds that can see UV light will be able to see this reflectance. And then courtship displays are sets of behaviors that a bird will do when trying to attract a mate. This often may include like a dance we're used to seeing in birds. And then during these courtship dances, the birds often show off particularly pretty and colorful feathers. To carry out this study, they examined 108 different bird species from Australia. They measured their UV reflective plumage using an optical spectrometer. Each reading was an average of 10 readings, and 5 birds were read for each species. They examined different wavelengths reflected in different body regions, including the head, throat, and nape, the chest, flanks, belly, and vent, the back and the rump, and the tail. In order to figure out if UV reflective plumage plays a role in sexual signaling, they looked at the correlation of UV reflective plumage with plumage areas used in courtship displays. The researchers gathered information from other studies about the specifics of how each different bird species behaved during courtship display, specifically which feathers they showed off to their potential mate. The significance of the correlation between courtship display plumage and UV reflective plumage was determined by binomial tests. The results show that UV reflective plumage was found in one or more body region in 72% of the bird species. Then 93% of these birds had UV reflective plumage in areas of the body that were used in courtship displays. This graph shows how other colors, such as the long and medium wavelength colors, were not found to be significantly more prevalent in courtship regions, whereas the UV colors were significantly more prevalent in the courtship regions. These results strongly suggest that UV reflective plumage plays a role in sexual signaling. It also tells us that bright, high-contrast colors in general are not significantly linked to sexual signaling, but rather these secret UV colors are much more significant in courtship displays and mate choice. But why should UV signals in birds be associated with courtship displays? We never really got into this, but these researchers made some predictions. So first is that UV may be a good medium for signaling over short distances because it is more rapidly degraded over longer distances than longer wavelengths due to particle scatter. UV signals can then be directed at intended receivers while remaining obscure to eavesdroppers. So the second one is where UV may constitute a secret avian channel for communication because many potential mammalian predators are unable to perceive UV light. And the third is that UV signals may be favored for signaling because they contrast strongly with background foliage. This occurs because chlorophyll in plants absorbs UV light rather than reflecting it. So most plants will provide a highly contrasting backdrop to the UV reflective plumage on birds. And lastly, UV signals may be sensitive indicators of the quality of the feathers. Many UV reflective plumage birds are created in part by the microstructure of the feathers rather than by pigmentation. So the strength of the UV signals may actually indicate the feather's quality.
To end off with some final thoughts, we learned that a wide variety of animals are capable of seeing UV light, with some being able to use it for communication. The specific examples being the UV reflectance of wing scales determining success of male mating in Coleus erytheme, and UV reflective plumage in birds playing a role in sexual signaling.
And that's everything we have for you today on UV light signaling in animals. Thank you for watching.
These are our first set of references used for this presentation, and these are the second set of references. Thank you.